Hi, this is Dr. Bilal Hussain. I'm the first year cardiology fellow at the University of Cincinnati. Thank you for joining us at ACC 25 in Chicago. I'm in Dr. Bhatt, who is the director of Mount Sinai Fuster Heart Hospital. Thank you for joining us, Dr. Bhatt.
Great to be with you.
He just presented his late-breaking trial on the REVERSE-IT trial, so we're here to dig more into it. Can you tell us a little bit more about it, Dr. Bhatt?
Yeah, absolutely. What the trial was testing was bentracimab. That is an intravenous monoclonal antibody that was designed to reverse the effect of ticagrelor. Of course, ticagrelor is an antiplatelet agent. Like all antiplatelet agents, it can cause bleeding. But one aspect of ticagrelor is that it is a reversible antiplatelet agent. That doesn't mean it's reversible in terms of it's gone in 5 minutes. Of course, its effect lasts for about 3 to 5 days. But the reversibility refers to how ticagrelor binds to the ADP P2Y12 receptor on the platelet. That's unlike clopidogrel or prasugrel. Those are irreversible antiplatelet agents, so their active metabolites bind to that same receptor for the lifespan of the platelets, 7 to 10 days or so. So quite different. That's why platelet transfusions really don't work for ticagrelor. But that characteristic of reversibility has allowed development of this monoclonal antibody. So what we tested in REVERSE-IT, well, just to go back one step, there's a phase 1 trial in healthy human volunteers, patients randomized to bentracimab or placebo. And there we found significant restoration of normal platelet function. That was presented as a late breaker. I presented that a few years ago at ACC and published in New England Journal of Medicine. So that's phase 1. Phase 1's healthy human volunteers. What I presented here, REVERSE-IT, was the pivotal trial designed for hopefully regulatory approval, the phase 3 trial known as REVERSE-IT. And here we were studying actual patients, either needing emergency surgery or with major spontaneous bleeding. So sick patients, for the most part, that we wanted to make sure that the platelet reversal that we saw, which was so effective in the phase 1, actually was also effective in real patients. And we found there was.
Okay, can you tell us about the comorbidities of these patients, and do you feel like if any of the comorbidities specifically affected the reversal you saw?
It's a great question. I mean, that was at least a theoretical concern. Maybe the drug wouldn't perform as well in actual patients versus healthy human volunteers. But in fact, what we found was significant restoration of platelet function and hemostasis as well. We assessed clinical hemostasis in the overall trial, but also in the 2 large subgroups of patients needing surgical procedures, and it was, again, urgent or emergent surgery, or patients that were having major bleeding episodes. So the results were significant in both those subgroups. In fact, in terms of restoration of platelet function, that was statistically significant in all our pre-specified subgroups. So really consistent results. That's the platelet part of the story, but we did also look at hemostasis. And this is a non-randomized study, so how do you assess hemostasis? It can be a little bit challenging to make sure there's objectivity to it, right? Because if you maybe ask the surgeon, is there hemostasis? Well, the surgeon might say, there's always hemostasis. So, there has to be some objectivity, but we had an independent clinical events committee, or CEC, adjudicating these events, chaired by Bob Giuliano from the Brigham. And they assessed the degree of hemostasis and thought it was effective in 100% of the surgical patients and 83% of the bleeding patients. And intracranial hemorrhage, those patients are perhaps the scariest to physicians. There we found an 80% rate of effective hemostasis as assessed by those independent adjudicators. So overall, it seemed to perform well. We didn't identify any major safety issues, no serious allergic reactions, no serious thrombotic events that we thought were due to the drug. Of course, there were some MIs and strokes. These are sick patients, some DVTs, that sort of thing. But in terms of serious allergic reactions, serious adverse events due to the drug leading to drug discontinuation or termination from the study, we actually didn't see any of those.
That's pretty good. There are no major adverse events, so I think that's pretty promising. Can you tell us about? I know that the bentracimab was given as a bolus and then a continued infusion for 16 hours. Do you think if the patient needs a longer reversal duration, can the bentracimab continuation past those duration be considered?
So just to make clear to everyone listening, this is an investigational agent, at least at the time of this taping, not approved by the FDA or any other regulatory agency. Usually those processes take some time and effort, but eventually the FDA I hope, will opine, and there'll be a specific bolus infusion and dosing and, labeling and so forth. But to answer your question, theoretically, yes, it could be possible to continue the infusion for as long as you need, for as long as you think there's still, ticagrelor activity that's on board. In theory, you could sort of even titrate the, length of infusion. So maybe you're in the cath lab, you have a perforation, you want to reverse ticagrelor because that's on board, but then you put in a covered stent and the perforation's okay, and then you want the patient back on ticagrelor because you just put in a rather thrombogenic, covered stent. So potentially that sort of flexibility will be there. I think that might take additional study though, because we studied it in one particular way, but there's a lot of potential flexibility. And we studied it, in sick patients, but in theory, this isn't what we studied, but one could consider elective surgeries as well, where, patient comes in, they're on ticagrelor, maybe they had a stent last week, now they've fallen, there's a broken hip, it's a little bit unstable, you don't really want to wait too long, might be bad for the hip, patient's in pain. You don't really want to keep them off ticagrelor for too long because they just got a stent. So there, you can imagine potentially reverse the ticagrelor, do the hip, and then as soon as the surgeon says you can resume the ticagrelor, resume it. So, but again, that's not what we studied. We studied people needing really urgent or emergent surgery or with major bleeding.
But definitely, if it can be used to, kind of minimize the unnecessary delay in the surgery which the patient needs, I think that would be like a pretty good use. I know you talked about some anti-drug antibodies in your initial publication, New England Journal of Medicine. Were you able to look more into it, or did you get any more data if it affected the PRU levels you were seeing?
Yeah, it's a great question. And of course, in this development program, we've looked at anti-drug antibodies, and that's not uncommon with drugs, but we didn't see any correlation. Well, we didn't see any serious allergic reactions. So, there was, I think, one minor skin injection site warmth or something, but nothing in terms of serious adverse events or allergic reactions. So it seems like the drug is pretty well tolerated. As far as we can tell, it would probably be okay to re-administer it. That's something that you might theoretically be concerned with. But on the other hand, I think it might be doctors that don't use a lot of monoclonal antibodies, like cardiologists that worry about that sort of thing, because there are other monoclonal antibodies that have been used in cardiology where re-administration really hasn't been a problem. So if you think about the PCSK9 inhibitors, at least two that are approved, and monoclonal antibodies, obviously there's redosing that's occurring there all the time. I think with this compound, from at least what we could see in the phase 3 trial, 226 patients, so modest sample size, but at least within that context, didn't seem to be an issue.
Okay, and I know the FDA just designated the drug as the orphan drug status last week. Congratulations for that. And what do you think the challenges you might have for the final regulatory assessment by the FDA? Well,
that's a tough question. Ultimately, it's the company, in this case or in all cases, that submits the data to the FDA and interacts most directly with the FDA. So have to see how all that goes. Really, the investigators have sort of done their job, I think, with conducting a really good trial in a challenging group of patients. Not easy to enroll patients, right? Someone comes in with an intracranial hemorrhage on Saturday night. It's hard enough to take care of them, but then to enroll them in a clinical trial, that's one of the reasons, at least our investigators didn't feel that a randomized design was going to be feasible. Obviously, randomization, I'm a big believer in it in principle, but at least our particular site investigators in this trial didn't think, they didn't feel comfortable randomizing because they knew from the phase 1 data that the agent worked in terms of, restoring platelet function. But to answer your question, though, yeah, it'll depend on the FDA's review of the data. It's not uncommon, that they might want additional information. That's typically the case.
Okay. So my final question, what advice would you give the residence fellows who want to be involved in groundbreaking trials like this, and especially how to go about collaborating with somebody who's going to be involved in designing these monoclonal antibodies and all these cutting-edge technologies?
Sure. I think even, for folks that don't necessarily want to have a research or academic career or aren't sure, always good to do some research just to, get a sense of what's going on as you use the word cutting edge. Now, because you never know exactly what you'll be doing in the future. Even if you end up in a private practice setting, a lot of the best sites when I'm doing trials are high volume private practices. They're oftentimes very efficient and well organized. So, even if you're not necessarily going to be a hardcore researcher yourself, it's good to have some background in that, and just to be able to keep up with things and interpret the literature. So I would encourage all the folks that are listening that are in training to take a bit of time to engage in academic activities and research projects. To answer your question concretely, one thing that I think is really useful is being a site co-investigator on a trial. There's not always a lot of academic credit. There's usually no academic credit for that sort of thing, but I know I did a ton of it earlier on in my career, and I found it to be really interesting, really educational. I mean, I remember enrolling patients in some of the older Gusto trials. Maybe the audience doesn't remember what those were, but those are the trials of, that put lytics on the map for ST elevation MI. Of course, now we're all doing primary PCI in most regions of the world, but, those were large trials, and you're not going to see my name in any of the publications anywhere, but, I was there, on Saturday nights, enrolling patients, consenting them, and doing that sort of thing. And I loved it. I thought it was exciting, but it was also very educational, and gave me a lot of insights into clinical trials and, what sort of patients get in, importantly, what sort get excluded, how generalizable are trials. So there's a lot, you can learn from textbooks and lectures, but there's a lot you can learn experientially in terms of research. So if you're not really in the thick of it doing it, you never have a really good sense of what it's about or whether you really like it or not. So I would encourage everyone to do that. It's pretty low investment in terms of career commitment to be a co-investigator in a trial. So I'd say do it. But of course, if you are, then you want to do a good job.
Yeah, for sure. Thank you for being here, Dr. Bhatt, and taking the time to talk to us about the trial. Appreciate it.
It's wonderful speaking with you.
Thank you. For more videos like this, check out our channel at youtube.com/FITsOnTheGo. Thank you.